So we're now going to look at something called the polymerase chain reaction, or PCR. It is possibly one of the most important techniques that have been developed in terms of using DNA in the last 50 years. It's how we do DNA fingerprinting, it's how we extract sections of DNA to work with. It's pretty important, and you need to know it in quite a lot of detail. Okay, so when you're working with DNA, the first thing you need to do is to get the bit of DNA you need to work with. So, I mean, if you decide you want to produce your one copy of the insulin gene, you're going to need lots and lots of copies of the insulin gene. So you can either cut out that particular section of gene from millions of cells and then gather together all those little bits, or, and this is a bit that's quite clever, you can take one, find out where it is, and just copy that section again and again and again. And that's what PCR does. So what you do and what you'll be using, and we'll talk about these a bit more as well when we talk about genetic engineering, you can cut DNA with endonucleases, and you should have heard them in genetic engineering. Endo is inside, nucleases would be nucleic acid, and it's an enzyme, so it's an enzyme that cuts inside nucleic acids. And ligase you have heard of, because that's joining together bits. So basically we can cut bits out and we can join them together. What you can also do, and is really important, is that you can separate out bits of DNA by size, and then you can also catch those bits. So that's called blotting. And again, we'll take you through how to use gel electrophoresis, but it's also in your notes. So PCR. So polymerase chain reaction, you amplify a specific sequence. Now that is really important. It's a particular bit that you're going to get, and then you're going to get lots of that particular section. What you use is a machine called a thermocycler. I mean, these have come on leaps and bounds. This is actually one that you would probably have. This is a bench one, and you would, if you go to uni and you work in like even a first year uni lab, you'd have one at the end of every bench. When I was doing my degree many moons ago, obviously, PCR was only really starting to get to the point where you could get things working really well, and these were super expensive. There was like 2 in the lab that I went and did my project in. So what you have here is basically inside this bit in here, you have a block of metal, and these little bits here, these tiny little bits here, are where you put little test tubes. Well, they're not kind of test tubes, they're called microtubes, so they kind of look like that with a little kind of lid on the top of them. And they kind of slot in to the metal block. And then in here, you spend a lot of time figuring out exactly how many cycles you want to do, exactly what temperature, and exactly how long you want it to be held there for. But we'll come back to that in a second. Okay, so this metal block basically can heat up and cool down really quickly in a very controlled way. So inside, that's a much better picture of a microcentrifuge tube. Okay, so what you have in here, you're going to need primers, because you can't copy DNA without primers. We know that from your DNA replication. You're going to need a DNA polymerase. But because of the way this works, remember I said we had a block that heated and cooled. Well, if it's going to heat it, I need something that's going to survive heat. Because normally if you heat up any kind of enzyme from like our cells, anything over about 40 degrees and you start to get a problem because it denatures. So you also need nucleotides. Again, not surprising. And you need to have other things that give the right conditions. So you get a pH buffer has to be right, and you need magnesium ions just to give everything what you need to run. Okay, so in normal DNA replication, you just need a load of primers that will allow you to get all the way up the strand. But in PCR, you actually need primers that bracket the sequence that you want. So if this was your whole chromosome, and sorry, let's make it double, because obviously they're double-stranded. Imagine that's a whole pile of neatly drawn in lines. Okay, and the section that you want is actually here. Well, what you're going to need to do is get a primer for up here and a primer for down here. because remember DNA polymerase can only work in one direction. So this is going to be the attachment for at 3' and that's going to be attachment at 3' on that side. So you need to know what your DNA is before you can actually find your DNA. So that's a little bit annoying. But we have full genetic sequence for quite a lot of species now, including humans, and we can use this really well. So what you then need is, once you've got your sequence data, you need the primers, and you can just order them. You get machines that can literally just churn out. You can call up and say, I need to have a ATGTGC primer, and it can just make that. And what you've now got is the sequences that you need to target either side of your section. Okay? Your DNA polymerase. So I say that has to be a bit special, because normal DNA polymerase, in terms of like for us, would not cope with PCR, because PCR is going to heat up the DNA to around about 95 degrees. So what they've done is they've gone looking for a DNA polymerase that can handle that, and where they found it was, not surprisingly, from some archaebacteria. These are bacteria that live in nasty places. We mentioned it right at the beginning for your prokaryotes. And they found Taq polymerase inside, because you can see Thermophilus aqueous, so that's the Taq polymerase. And they found these in thermal vents, they found them in hot springs, they found lots of these different kind of heat tolerant bacteria, and they basically just extracted the enzyme from them. So they now have vats that are producing these bacteria in the right temperature for them to be happy, and then harvesting the polymerase. So it has to be heat tolerant, as I say, because we are going to heat this up, and it's going to be cycles of this. Anything, as I say, normally would just denature. So what we're going to do is heat up the DNA, and you're going to basically snap the hydrogen bonds, because as you heat it, they are the weaker bonds, so they will basically melt apart. And once they've melted apart, your DNA strands separate. But you're not going to take it high enough that it's going to break the sugar phosphate bonds. So you're taking the temperature up high enough to break the hydrogen bonds, but not high enough to break the covalents. So yeah, in the 90s, so normal enzymes, what we would consider your normal meso enzymes, they're not going to be any good. So this is where you start. So first cycle of DNA, we have our normal DNA strand, so 5' at one end, 5' at the other end on the other side. Okay, so heated up between 92 to 98 degrees. That's pretty hot, but it's only for a few seconds. But what's going to happen is we are going to snap all of these bonds down here, but we're not going to break the backbones. So what we end up with is a separated DNA strand. Okay, so we now need to cool it down, because at this point nothing's going to work, nothing's going to stick, because what we need to do is now stick our primers. Right, this has been very abridged, so this would actually be several bases long, not just 2. Okay, so we have to cool it down, as I say, 50 to 65 degrees, because you're going to need to know the numbers, unfortunately. Okay, so if you cool it down to that, the hydrogen bonds can reform now, so we can stick in our primers and they will attach. Now, we need to heat it back up again, but this time we're going to 70 to 80 degrees. So we're going up to 70 to 80 degrees because that is the optimum, important, for the Taq polymerase. So at the optimum for that one, if we left it at 50 to 55, it might have done some work, but it would take forever. If we heat it much more than this, it will just stop working, but it won't be destroyed. But anything higher than about 90, well, you're going to melt back off your hydrogen bonds, so you just break apart any strand you want. So this temperature is really tricky to get right, and you have to get it perfectly right. Okay, and then these strands will form. You now heat it back up again. And when you heat it back up again, we split our new ones that we formed from our original ones. Okay, and then we cool it back down. And our primers now stick in on our new strands and our original strands. And now we heat it back up. When we heat it back up, we get to our correct temperature for the polymerase, and we now get all of our new ones forming. And you keep doing this again and again and again in cycles. So a diagram showing the kind of overview of the whole thing. So we start with our original DNA strand and we denature that just with heat. So in the 90s, you then cool it back down to allow the primers to anneal. That is the term you need to use, means stick to complementary parts. And you then, once they're annealed, you then need to bump it up to the optimum for the DNA synthesis. So that's using TAC, so it's going to be up in the 70s. And then once you've got the full length of this, now again, getting that length, like how long you leave this for. Remember, the DNA polymerase is going to work its way along. If you leave it for not enough time, it's going to not have the full sequence. If you leave it for too long, you end up with extra bits you don't really want on your sequence, which can blur the kind of edges of what you need. So you need to actually work out exactly how long you want it to be working for. And then finally, you just restart again, and you keep doing that. So this is your potential amplification. So you start with 1 copy. That 1 copy goes to 2. 2 copies make 4, 8, 16, 32. You should be used to this kind of number set at the moment, because this is what they're talking a lot about in terms of corona. This is an exponential growth curve. It's a doubling curve. So this is why, in terms of corona stuff, we want the R number to be much, much less than 1. Anything greater, equal or greater to 1, and we have this curve starting again. And so you can see, if you run it for 20 cycles, you can actually have, well, what have we got there? Over 1 million. You know, so you copies of your particular target sequence that you want. It's an amazing way to do things. So why would you even want to do it? Here are your examples of reasons. Okay, DNA profiling. So that would be either individuals that you need to look at specific reasons for that, or familial things if you're looking for, like, literally DNA fingerprinting for paternity testing, things like that. Population studies, looking for how frequent a particular gene is or a particular allele is within a gene. Gene detection for disease diagnostics, so checking for specific genes that you know are increased risk for particular genetic disorders. Forensics, so that could be for crime, that could be for archaeology, so working out what something was in terms of tracking evolutionary stuff. So this is a animated version of the PCR. I think it's actually quite a nice one. This will run through, but obviously I will give you this as a separate video file and you can rewatch it a couple of times if you want to.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme TAC polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.